So beta-plated sheets are another example of a secondary structure of proteins. Remember that we have two different types of secondary structures. We have the alpha helix and we have the beta-plated sheet. So important keynotes to make over here is that we still see that planar character at our peptide bond, because remember that these electrons over here are delocalized and they're shared between the oxygen and the nitrogen. So this nitrogen is actually has a double bond character with the carbon. Now, this delocalization generates that partial negative on oxygen and a partial positive on the nitrogen, and these partial positive and partial negative charges are what contributes to the hydrogen bonding in our secondary structures. It's important to note that in our secondary structures, the R groups are not interacting through hydrogen bonding. It is our oxygen and nitrogen that are interacting via hydrogen bonding. So here we can see an oxygen. So this oxygen is negatively charged and it's interacting with this hydrogen right over here in white, which is bound to the nitrogen. So this nitrogen is partial positive, oxygen partial negative. So this oxygen is attracted to this hydrogen over here. And this is all due to that delocalization of these electrons and that partial double bond character. Now, let's make note that we have an anti-parallel and a parallel sheet. Both of these are common. The only difference is that here in our parallel sheet, whatever we are starting with, it's the same for each strand. So in each strand, we're starting with the amino terminus, ending with the carboxyl terminus. And we still have those interactions between the nitrogen and the oxygen. Here, it's the reverse. So if we're starting with an amino for this strand, we're going to start with a carboxyl group on this end. So it's the reverse. Here we can actually see that the terminal ends, so our carboxyl and amino terminal ends, are actually able to interact with one another, but here they are not able to interact with one another because they are not directly opposites. Now, the last factor that contributes to our beta sheets is R groups of amino acids. So we can note in purple over here, these purple groups are actually the side chains of our amino acids. Now, we don't want these side chains to be too big and bulky because look how compact this beta-plated sheet is. If we add a really large R group, we can imagine the steric hindrance it would cause, and it would actually disrupt the beta-plated sheet structure. So here on our alpha carbon, we know we've got the hydrogen, and let's say we have a really big R group, it's going to disrupt that chain below, and that is not favorable. And that is essentially our beta-plated sheets. We have that planar partial double bond character, which contributes to the partial negative and partial positive charges, which allows us to take part in hydrogen bonding. And we have anti-parallel and parallel beta-plated sheets, which are both common. The only difference is the route that they are taking. And lastly, we cannot have groups that are too bulky. We prefer, in beta-plated sheets, it is preferred to have smaller groups like serine or threonine, alanine, amino acids like that have smaller R groups. And lastly, what we should remember is that beta-plated sheets are secondary structures where the hydrogen bonding occurs on the backbone peptide bonds and not the R groups. That is very important to remember. Well, one last thing to point out is that remember that on our alpha helix, when we had our alpha helix, the hydrogen bonding was occurring between every 4 amino acids. But over here on the beta-plated sheet, we can see that the hydrogen bonding is occurring next to adjacent residues. So here it's adjacent residues interacting, but on our alpha helix, it was every 4 residues that were interacting. So the alpha helix, beta-plated sheet, both examples of secondary structures of peptides.